Thank you. It's my pleasure to share some of our work in laboratory with this audience. And my talk will cover two areas. Basically, it's surrounding the old structure that's been known for a century, bacterial capsule. And first, I will talk about how bacteria use the capsule as a shield for survival in the human host, particularly when they enter our blood circulation. And so the next part is the main part that covered with the title in the book, is how vaccine works based on the polysaccharide vaccine, polysaccharide materials. Okay, so I think Cesar already gave a very nice introduction of how bacteria impact our human health, and I'm not going to labor through the numbers, and we all know that bacteria is important. And for all the bacteria that cause the human mortality, and there are four or five bacteria majorly responsible for the death. You notice the four of them, four of these bacteria carries this so-called capsule. This is a streptococcus pneumonia with a polysaccharide capsule. This is a cell, bacterial cell, and the outside of structure is the polysaccharide capsule. And so it's a really thick layer of structure. And so in terms of human infection, bacteria can infect almost everywhere in our body. I think we are just a candidate for food. If you allow the bacteria to survive and replicate, they will do. And so most of the infection occurs with the primary side, and they are mostly limiting, self-limiting. However, when the bacteria transfer to the typically sterile side, they will cause the so-called invasive infection, for example, lung, blood, and brain. Okay, so the bacteria capsule has a lot of important features. For example, this is a light microscope. You see this white bar is actually bacteria, but the halo is bacteria. So the capsule is bigger than the bacteria itself. And so this is the picture I just showed you with the ceram pneumo. And so, so the bacteria, once they get into the typically sterile site, they will replicate and induce inflammation, and most of the death caused by septic infections. And so capsule is very famous because it's important for pathogenesis, and also capsule. So my scientific grandpa, Avery, has made a great discovery of DNA as the material for genetic material and in 1944. And so all the capsules are different in terms of structure, but they have some common features. All the capsules, most of them made of polysaccharide repeat units. Here are some examples, with one exception, the Bacillus atheraceus. That's a favorite organism Terry has studied for many years. It's made of made of single amino acid, but that's really rare. Virtually all the other polysaccharide, the capsule is made of polysaccharide. And I think we can debate why not all bacteria use one material or use the half or half. Okay, so the polysaccharide capsule is made from these capsule biosynthetic machinery. So in gram-positive bacteria, it's simpler because it only have one membrane. So polysaccharide is purified. Initiate synthesis in the inside of cell, and then once I have a repeat unit, flip over and elongated to a point that will anchor to the cell wall. For gram-negative bacteria, it's more difficult because it has to go through two layers of membrane. And so, but the basic feature of the gram-positive and negative bacteria capsule biosynthesis machinery is highly similar. Okay, so how capsule works? So with the knowledge of capsule for so many years, there's some evidence that how capsule works. However, most of the data have been generated in vitro experiments. So lots of in vitro experiments show the bacteria with capsule, it's slippery, does not bind to cell well, does not bind to any molecule very well, because it's negatively charged, and our cells are mostly negatively charged. And so there are some limited in vivo data show that capsule can, once we encapsulate the bacteria, once they get into blood, and they will survive, and without capsule, they won't. So the paradigm is the capsule protects the bacteria from phagocytic killing. That's absolutely right. However, the last part is that it happens in the spleen or the blood circulation, because we have a lot of phagocytes in the spleen and also with some phagocytes in the blood. Okay, however, after search many old literature, this is an example that shows that once the bacteria get into blood, they will end up in the liver, right? So they're not killed in the blood immediately. So with the limit of time, I'll just show you directly how we find that actually liver is the bacteria capsules target for immune evasion. And so this is a model we used the mouse model. Basically, you used IV inoculation of bacteria through a tail vein, and use the retrooperative bleeding to figure out in every single mouse and how bacteria disappears. So this is without capsule, the bacteria just disappear very quickly. With capsule, the bacteria sustain in the blood. So if you track where bacteria goes, so without capsule, the bacteria will be trapped in the liver. This is the yellow section if you count bacteria in all the major organs. And so they are not dying in blood, they're trapped in the liver. However, with capsule, and the bacteria will end up in the blood, right? And so, so let's just watch a movie to see how that occurs. So this is so I'm going to use the intravitreal microscopy imaging to show you how that will encapsulate bacteria and unencapsulated bacteria end up in the liver. And so this is a liver sinusoid structure. It's very simple with a layer of endothelial cells and also embedded Kupffer cells, which is the major residential macrophage in the liver, which consists of probably occupies about 90% of all macrophage we have. Okay, so on the left, that's unencapsulated bacteria with green. You will stain the blood vessel of the liver with blue, and so red is the macrophage. You see that without capsule, the bacteria will stick to the macrophage very quickly. However, with let's see, one more time. With the capsule, this bacteria will go through the liver like a superhighway, right? So liver is really the major organ for immune evasion of the capsule. So since this is already being published, so I just summarized here that so when bacteria in the blood, if they do not have a capsule protection, they will end up with the Kupffer cells get killed. And if they have capsule protection, and they will survive. However, I don't have time to show you that different capsule has different capacity to evade the binding to Kupffer cells. And so that explains why there's a great difference in serotype-dependent virulence. So not all capsule types are equal in terms of evading this immune antibacterial machinery. So I would be happy to talk to you after the lecture. Okay, so let's skip the gear to how the bacteria capsule is used as an effective vaccine adjutant. Okay, so as you know, the invasive bacteria, a few invasive bacteria, Staph aureus, Haemophilus, Neisseria, and Salmonella typhi, their capsule have been developed into an effective vaccine. Okay, so there are two polysaccharide vaccine forms. One is just a pure polysaccharide. Okay, so the purified capsule is mixed with the alum adjuvant and given to human, and so it's only induced the B cell, only active B cell without engaged T cell. So therefore, mostly it produced the IgM and IgG2 in human. However, in the new generation of polysaccharide vaccine, it's conjugated to carrier protein, mostly toxoid, or the proteins that has a strong immunogenicity. And those antigens can be delivered to T cell, and so activate T cell, B cell same time, and that will engage you activate the B cell to produce IgG isoforms, which are better antibacterial form of antibodies. Okay, so there are a lot of vaccines that target Strep pneumo, and so I've just used the Strep pneumo example here to show you how a polysaccharide vaccine works and how the conjugated vaccine works. Their mechanisms are very different. Okay, so here are, PPV23 consists of just pure polysaccharide from 23 serotypes of Strep pneumo. And so this has been licensed many years ago. They're probably the oldest form of polysaccharide vaccine for Strep pneumo. However, in 2000, the first conjugated vaccine induced in the United States as a PCV7, and then it's gradually increased with more serotype coverage. Now, Merck just produced a 21 serotype vaccine this year. Okay, and in terms of business, the polysaccharide vaccine, the PCV13 produced by Pfizer, sells the best in all the drugs, not just vaccine, all the drugs. This is the bestseller drug in the world in 2022. I'm not sure what happened in the last 2 years, but this is the very good or business model. Okay, so vaccine worked quite well, and so you have vaccine, and then certainly you need an antibody, and so we have all the ways to formulate addition to produce a good amount of antibody, and then you're looking for protection, right? However, we don't know much about how from antibody to protection. And we assume that bacteria, once you have antibody, the bacteria will be labeled by antibody in the blood, and then will be killed in the blood, okay? And then let's see if that's the case. All right, so here I'm going to use the two vaccines. One is the polysaccharide, PPV23, and the other one is conjugated vaccine, PCV13. These two vaccines made by the same company, right? And also they share 12 same polysaccharide, the 12 serotypes in terms of their antigen. So we can directly compare their effect with the same antigen in different forms, right? Okay. So first we use the mouse model to test how good is the conjugated vaccine in mice. It's very powerful, and so it offers almost 500,000-fold of protection. This is the best protection I have ever seen in my life in terms of vaccine potency. Okay, so how that happens? So we used our mouse model to look at how the bacteria, once they get into blood, how the bacteria behave. Okay, so without blood, without immunization, the bacteria stay in the blood, but immunized, the bacteria will decay from blood very rapidly. However, they are not dying, and so the vaccinated mice will trap all the bacteria, most of the bacteria in the liver, right? And the bacteria will be killed if you waited long enough. Within 30 minutes, almost all the bacteria is gone. So liver is very good at capture bacteria and kill bacteria in the immunized host. Okay, so the one wild experiment we did was to see if the effect sizes are required for the protection, because that's what is in the textbook. So we did one experiment, and we delete all the major effect sites with collagenated, we depleted all the macrophage. With the GR1 antibody, we depleted monocyte and neutrophil. And see, that's after immunization. This experiment shows that if you deplete all the major effect sites, the mice still alive. So this is unimmunized mice. These are the normal mice after immunization, and also the mice that lack the major effect site. So what's happening? So that was really surprised because I have a very limited knowledge of immunology, and I thought the effect sites take care of everything. Okay, so if you look at the bacterial decay, And the same thing, that if you, without major phagocytes, bacteria also disappear from blood with some mild deficiency, but anyway, within half hour, all the bacteria is gone. And so if you follow bacteria in the liver, and without phagocytes, the bacteria also localize in the liver, or they are killed in the liver. And so let's see where they go. Okay, so I just showed the movie. On the left is unimmunized mice, and with same type of IVM. You see, this is right. It's PCV immunized mice. The bacteria either stick to phagocyte, to macrophage, but most of them will stick to the blood vessel. Okay, and some of them clump, get big clump, and the famous neutrophils, you can see with the purple, they come over and they go, right? So that means neutrophil is not a major phagocyte that kills bacteria in the blood. It's the liver, and it's the liver endothelial cells. Okay, so that's quite a surprise. Okay, so we then depleted the macrophages to see if endothelial cell can directly capture the bacteria. And so this on the right, this is a mice without liver macrophage. The bacteria captured by endothelial cell quite well. So that means endothelial can do bacterial capture with the after immunization. Okay, so this is just quantified results, and for without immunization, the bacteria circulating in the blood, and if you immunize, then most of the bacteria will end up in endothelial cell, some of them on the macrophage. But without macrophage, endothelial cells still can capture the bacteria. Okay? All right. So what happens? Do we need an antibody for this? And so yes. So if you so in mu-MT mice after immunization, nothing happens with the so bacteria still persistently in the blood. So we need the antibody for liver capture of the bacteria. And make long story short, through various methods, we identified the low affinity IgG receptor, Fc gamma 2b, as a receptor on the endothelial cell that is responsible for capture of the antibody-coated bacteria. So this is a wild type. It has a lot of bacteria captured on the endothelial cell. If you knock out Fc gamma 2b, the bacteria only macrophage can capture bacteria, not endothelial cell. Okay. So let me summarize this part. So if you have a PCV or conjugate vaccine immunized mice, they produce the antibody, IgG antibody. This will label the bacteria and it will end up attach bacteria to endothelial cells predominantly, and some of the bacteria also will attach to Kupffer cells. The bacteria will be killed in the liver. Okay. Now let's see what happens with the polysaccharide vaccine, because it has been used in senior individuals in the States and many other countries. Okay, so we did a similar experiment in mice. That make long story short, this polysaccharide vaccine has a certain level of protection, but the protection level is way low than conjugate vaccine. It has it's about 400-fold lower than conjugate vaccine. Okay, so I think this is the first time that there's a study directly compare the conjugate versus the unconjugated polysaccharide. Okay, and so the polysaccharide vaccine can also activate the liver to capture the bacteria, and also to kill the bacteria. However, this is with a low dose of infection, but if you increase the infection dose, the polysaccharide vaccine no longer works. Okay? Right, phagocytes is very important. So if after immunization, if we get rid of phagocytes, the mice die very quickly, regardless if you have immunization or not. And so without phagocytes, the polysaccharide vaccine immunized mice cannot capture the bacteria in the liver. Okay? All right, so this is the IVM. I'm going to just skip this because we saw this before. And antibody is also very important. So polysaccharide vaccine immunized, basically in mice, mostly produce IgM and very barely produce IgG. Okay, and so in immune mice, without antibody, the protection doesn't occur. Okay. Okay, and then also we found that complements are essential for this polysaccharide-mediated capture in the liver. So in C3 mice, C3 is a key component of a complement system. Without C3, the immunization did not result in any bacterial capture, clearance in the blood or capturing in the liver. So therefore, only polysaccharide will induce in mice, at least this is in human also mostly true, produce IgM antibody. The IgM antibody does not have receptor, at least in mice, it does not have Fc receptor. And so that only after IgM attached to the bacteria, this will activate complement C3, and the C3 will deposit on bacteria, and that will allow Kupffer cells or macrophage to capture the bacteria, not endothelial cells. So you only activate the one type of cell in the liver. Okay, so the question is if this is only work for single bacteria or it's true for all the encapsulated bacteria. And so use the Neisseria meningitidis, which has a conjugate vaccine, and we basically repeated the same phenomenon. And so with the conjugate vaccine covers the two serotypes, it binds to the liver of the liver cells, endothelial cells bind bacteria well. If you get rid of the Fc gamma 2b, and then binding only occurs with the Kupffer cells. And then we move on to other encapsulated bacteria, for example, like Klebsiella pneumonia and other E. coli. Basically, we used, here we did not do conjugated vaccine versus the polysaccharide vaccine because there's no such a vaccine. And so we used the homemade whole cell vaccine. Basically, we just inactivated bacteria, immunized. It produced a certain level of low level of protection, and still it can induce the clearance of low infection dose in the blood, and also can capture the bacteria in the liver. However, it cannot activate high-level bacterial binding by endothelial cell, and so only activate Kupffer cells. So therefore, this protection is really low. And however, I think for many drug-resistant bacteria like Enterococcus, not Enterococcus, I guess the Klebsiella and E. coli and other encapsulated bacteria, I think a capsule-based conjugate vaccine will be one option to prevent the infection. Okay, so if this is relevant to humans, and so we did use the human that immunized by PCV13, used the serum from a human, did a similar treatment, passive protection experiment in humanized mice. These mice lack their own antibody receptor, IgG receptor, but they have all the major human IgG receptors knocked in. And so basically we recapitulate the similar finding with the humanized mice and a human antibody. Okay, so let's summarize. So we discovered this liver-based vaccinology phenomenon, and and I think that explains to a certain level why some vaccine works better than others. And so, for example, here we show that with the polysaccharide vaccine only activate Kupffer cell immunity, so it gives you a low level of protection. However, if the vaccine can activate both endothelial cell as well as Kupffer cells, then it will yield a high level of protection. I think this has some implication in the future how we can make a better antibody than others. Okay, so last couple of minutes, I'm going to show you what we can use this for vaccine development. So there's a bad need for reliable vaccine efficacy correlate in many pathogens. As I show, we can use the formulation, adjuvant, and other different ways to raise the antibody level. However, we don't know if sometimes the antibody is there and no protection. And so we just don't know what happens. It works, if it doesn't work, we don't know why that doesn't work. So I think some of the parameters I just showed you can be quantifiable or correlate in reflecting the in vivo vaccine immune reaction in the liver. And here, I'll just show you very briefly the work just published actually in vaccine done by Xianbin and Jing-Ren. And so we used pathogen clearance from blood and also the level of bacterial trapping in liver as well as the distribution of bacteria in Kupffer cells versus the endothelial cells. And we showed that we use a different dose of bacterial infection in a single vaccine. We can separate the efficacy of different serotypes. Okay, and so this is also reflected in the amount of bacteria that are trapped in the liver that are infected by different serotypes. Okay, so let me quickly summarize that I just show you bacterial capsule targets the liver macrophage for immune evasion. Liver mainly execute vaccine immunity against the invasive bacterial infections. So vaccine efficacy is determined by antibody-mediated bacterial capture, a low-level immunity by Kupffer cell alone, and a high level of immunity is achieved by a combination of Kupffer cells and endothelial cells. And here I show you the two types of vaccines, conjugate vaccine versus polysaccharide vaccine alone, and I think that's the only examples. And finally, liver-based vaccine mechanism, I think it can be used to develop the future vaccine with a better efficacy. So finally, I need to thank the hardworking students in my lab, and also a lot of collaborators. I don't have time to show all of them. Some of the major contributors, the students that are responsible for the discovery already show their picture in the PowerPoint. But anyway, thank you all.
Okay, very interesting story. We still have 2 minutes for questions. So there are microphones on both sides.
Okay.
We can see the bacteria, clearly see the bacteria attached to the endothelial cells of the liver. And I'm curious, is there a viability tracer that can be added to the bacteria that show that bacteria really killed by the endothelial cell? Or it's just attached there and there's some other players, for example, neutrophils that be recruited and then kill the bacteria?
Well, so neutrophils is not responsible for the killing because we demonstrate that in mice lack neutrophils and macrophage. However, the endothelial cell, so this is still in the progress of research. So we don't know how the bacteria are killed on endothelial cells. However,
so the bacteria is labeled by GFP, right?
Yes.
So
yes.
I think there are some fluorophores, for example, DS-red, you can label that and then you can show the viability of the cells and maybe you can
Yes, we did that. So we show that bacteria can, we actually did an in vitro experiment with isolated endothelial cells from liver of mice, and also we have a cell line from human. So in vitro, we can demonstrate bacteria, once they bind to endothelial cell, they will be killed. Some of the bacteria, very few, will end up in the cell. However, most of the bacteria actually killed outside of cell. This process absolutely requires complement C3. So I just showed you the conjugate vaccine does not need complement to bind, to deliver to the liver endothelial cells. However, if you get rid of C3, the bacteria will just stick to the endothelial cell, they will not die. So we don't know how C3 works to kill bacteria because this is I show you this gram-positive bacteria has a thick cell wall and not supposed to permit the formation of a membrane attack complex will kill bacteria. And so this is a very, very challenging question, and we are working on it.
I'm
Yeah.
really looking forward to the follow-ups.
Thank
Thank
you.
you very much.
Okay, we still have time for one more question.
It's really nice work. So I'm going to expose my immunology ignorance here and ask you, is there something special about the IgG receptor on liver endothelium as opposed to other endothelial cells in terms of concentration or function?
Great question. Yes. So I skipped a lot of details because of time. And so FT gamma 2B is known to be the inhibitory receptor on B cells. And the other places that express the FT gamma 2B is the liver of human and mice. So this receptor is exclusively produced in liver and not in other blood vessel. So that's why if you give the antibody labeled bacteria in the blood, the bacteria automatically will attach to liver cells instead of other blood vessels. So not all blood vessels are the same.
Thank you.
Okay, let's thank Professor